bacteria, primarily to test several lab-designed hydrogels and secondarily to explore the potential applications of hydrogel systems in biotechnology, drug delivery, and environmental remediation. In my methodology, I began with testing 25 different hydrogel formulations. I then narrowed them down to three based on their compatibility with the encapsulation protocol I used, which consists of submerging the hydrogel already mixed with bacteria in the cross-linking agent, calcium chloride in this case. These three formulations, F1, F2, and F3, had varying gelatin and sodium alginate concentration, respectively 2% sodium alginate, 2% gelatin for F1, 4% sodium alginate, 4% gelatin for F2, and 3% sodium alginate, 10% gelatin for F3. I encapsulated 4 bacterial strains, E. coli, Pseudomonas aeruginosa, Salmonella enterica, and Staphylococcus aureus within these three hydrogels. I monitored their viability using the optical density at 600 nanometers measurements and as an objective method to estimate the number of bacterial cells, and then assessed their metabolic activity using a resaccharin metabolism test, which is a second and indirect outcome measure consisting of detecting the fluorescence in the resaccharin product after metabolization over time. These tests were conducted at 3 different time points, 24, 48, and 72 hours post-encapsulation. The first part of my results focused on bacterial viability using the OD600 method through time. At the 24-hour time point, I observed a significant decrease in viability for the F3 formulation compared to the non-encapsulated controls. In contrast, F1 and F2 showed no significant difference in viability compared to the non-encapsulated controls. However, at both 48 hours and 72 hours, there were no significant differences in overall bacterial viability between the hydrogel formulations and non-encapsulated controls. In the second part of my results, I assessed the metabolic activity of bacteria in F1, F2, and F3. At 24 hours, there were no significant differences in metabolic activity between the hydrogel formulations and the non-encapsulated controls. However, at the 72 hours time point, F2 and F3 showed a significant increase in metabolic activity compared to the non-encapsulated controls, while F1 displayed no significant difference. The discussion highlights the importance of tailoring hydrogel formulations to the specific requirements of each bacterial strain to ensure not only their survival but also their metabolic capability. Encapsulation can provide a protective environment for bacteria, shielding them from adverse conditions and promoting their survival. Furthermore, the compounds forming the hydrogel, like gelatin in this case, can be a source of nutriments. In conclusion, the optimal hydrogel formulations for bacterial metabolic activity seem to vary between the tested bacterial strains. Further optimization and testing for hydrogel compositions may be required to achieve the best outcomes for each bacterial species. The viability and metabolic activity tests conducted during this study do not always show complete agreement, highlighting the complex nature of bacterial behavior within hydrogel formulations and the importance of using multiple assessment methods to evaluate their performance. The potential application of these hydrogel systems in biotechnology, pharmacology, and environmental studies should also be explored, as these strains, if modified, can be used as biosensors, bioremediation factors, and even highly sophisticated drug delivery systems. Future direction for this research include investigating the long-term effects of hydrogel formulations on bacterial survival and metabolic activity, exploring the specific mechanism underlying biofilm development in encapsulated systems, and optimizing hydrogel formulations for controlling bacterial growth and biofilm formation. Finally, I would like to express my gratitude to all my instructors and my classmates for their support and collaboration throughout this research projects. Thank you.